Hello students, we have been discussing about the plant growth till now. Now we'll start from the very beginning, that where from that particular growth starts from. And when we talk about growing a plant, what thing comes into our mind is that it must be a seed in the beginning, or it might be a vegetative part. But because we are considering over here the seed and seed germination, so we are going to take into account that a plant must have reproduced sexually by the process of pollination, where a pollen would come and bring the male gamete to the female gamete. The fertilization would take place inside the ovule, inside the ovary, and that ovule would later on form the seed. So imagine that I am having a seed in my hand and I want to sow that seed. From that point, from sowing that seed, the plant growth actually begins. So because we are dealing with the plant growth, so we are going to see that how is that the plant from the stage of seed undertakes the growth processes and gives rise to a big plant. Firstly, it is sapling in the beginning, then it grows, then it shows secondary growth. So we are going to deal with the growth step by step. That is, firstly we discussed that what are the factors which influence the growth, what is growth basically. Now we start from the seed and we will reach till the secondary growth. We are going to discuss each one step by step. So first of all, you need to understand that every plant somehow spent some stage in the suspended stage. When I say suspended stage, I mean to say that plant growth has stopped. There is a stage of suspended growth inside the plant, and that is seen inside the seed. Once the seed has been formed, it enters into the stage of quiescence. Now we know this word, it is an English word which tells that something is silent, that no activity is taking place. Now what you need to understand, that the suspended growth of a plant that is in the form of seed, the seed stage of the plant is in the suspended stage. It has to reach the seed germination, where it would give rise to a small plant that is known as a sapling. Now this suspended growth is in two forms, quiescence and dormancy. What we deal most is with dormancy. Now you need to understand the difference between quiescence and dormancy. While quiescence is exogenously controlled, dormancy is endogenous. Now I'm using the term exogenous and endogenous. That means supposedly the seed is in a quiescent stage, the seed will not develop because the external conditions, that is the external factors which are favorable for the growth, are not present. And in the case of dormancy, though the external conditions would be favorable for the growth, seed will not show germination. I'm going to repeat it. In case of quiescence, the external conditions are not favorable for the seed germination, but in case of dormancy, though the external conditions are favorable, like that for germinating seeds, all of you must be consuming sprouts. You put the seeds in water for some days, 1 or 2 days in moisture condition, and they show sprouting. That sprouting is basically seed germination. So if the seeds are in the quiescent stage, as soon as you give them watery condition, that is the moisturous condition, they show sprouting. That is, they were in the stage of quiescence. But you would find certain seeds that even after being put in the water or the condition of moisture and ample oxygen, they do not germinate. That means the control of germination was endogenous. Something inside them was responsible for not giving rise to the sprouting process. That particular condition is known as dormancy. And most of the seeds I'm telling you, they show suspended growth, okay, in the form of dormancy, that they enter the stage of dormancy and then either it is mechanically or chemically broken down. Alright, now we need to understand that quiescence is totally external conditions which are controlling quiescence. As soon as the external conditions are favorable, you're going to see the sprouts. But in case of dormancy, because it is endogenously controlled, so there must be few factors which are behind that dormant condition of the seed, and those conditions are, because the seed houses the embryo which gives rise to the new plant, the embryos which are present inside the seed, a dormant seed I'm talking about, it could be a rudimentary embryo. That means it may not be completely developed embryo, it cannot give rise to a new plant, it may not have the radicle or the plumule which gives rise to the shoot and the root system. respectively. The rudimentary embryos could be the one reason for dormancy. Apart from that, you know that seed coat is made up from the integuments of the ovule. Even if you don't know, just remember it for your learning purpose, that ovule gives rise to the seed, and ovule in the beginning has its coatings which are known as integuments, which get converted to the seed coats. The seed coats are very hard, you must have observed. So the seed coats, if they have become impermeable, that could be the reason for dormancy. Why I am saying that impermeable seed coats could be the reason of dormancy? Because for seed germination, you need the water to penetrate inside the seed, okay, to bring about the polysaccharide breakdown and other processes that we are going to discuss right now. Impermeable seed coats would not allow moisture and air to get inside the seed, and when that is not able to go inside, how do you expect the germination to take place? Then there is physiologically immature embryos could be there. First case was rudimentary embryos. That embryo might have not been formed, or it must be in the form of its cellular composition that is quite earlier, present in quite earlier stages of the embryo growth. Other is physiologically immature embryos. The embryos might have developed, it might have shown that growth, the endosperm might be present, but the physiological activities inside the embryo might not have taken place properly, due to which they stay immature. Alright, that would also not give rise to seed germination, and hence the seed would be termed as a dormant seed. That is, it is going to stay in suspended growth until and unless we apply some external mechanical or chemical pressure or some dormancy breaking method. And the last one is presence of germinating inhibitors. Now, these are the internal factors. In certain cases, seed shows the dormancy because in its proximity there are certain germination inhibitors. They are nothing but the chemicals which do not bring about the elongation of the radicle in the plumule, that is the breakdown and the other processes which are involved in the seed germination. The inhibitors are abscisic acid, which is responsible for making the seeds dormant. Then there is phenolic acid, which is again a germination inhibitor. Few short chain fatty acids are also responsible for germination, inhibiting the germination of the seed and keeping it dormant for a longer period. And a compound known as coumarin is also responsible for the inhibition of germination process. So this was all about dormancy. As soon as this dormancy or the quiescent stage is broken down, we are going to expect that the seed would germinate. Now let us see that what this germination is all about. Seed germination is all about. You know, let us take the example of a dicotyledonous seed that we take into consideration. These are the cotyledons. I'm not taking into detail the structure of seed over here, because here we are more concerned about how it germinates, alright, and what are the factors, how we see it from the growth point of view. That is important to see over here. Over here we have a small part which is part of embryo, that is known as radicle. This would give rise to the root, and the lower part, that is the plumule. Okay. This plumule later on gives rise to the shoot tip. This radicle gives rise to the root tip. As soon as the seed gets germinated in the soil, over here, this is the seed supposedly. The radicle goes downwards, the seed goes downwards, and the sapling comes outward. This is how the germination takes place. Alright. Now how the germination takes place, let us see. Let us see the structure of seed again in detail. Supposedly I am taking over here a gram seed. You must have seen gram seeds in your house. This looks like onion, more of an onion, but still I am considering it is as a gram seed. There is a small pore-like structure known as hilum over here. This hilum, we did consider that impermeable seed coats. This hilum is responsible for entry of water and oxygen into the seed. Now the cotyledons are rich in the reserve food material. As soon as the water enters by the process of imbibition, imbibition is a physical process where water would grow go across the diffusion gradient. That means this place is totally dry, and when it comes in contact with water, the water is bound to move inside the seed. It makes the inner part, which is rich in polysaccharides and other proteins, it makes the inner part rich in moisture and oxygen, and various physiological activities start taking place inside it. As a result, the volume increases and the seed coat breaks. Now, it is not supposed to break the entire seed coat. What it is supposed to do is that the cotyledons inside it should be able to generate such a hole so that the plumule and the radicle, they come out, and further they would show the growth. They would take up the material from the cotyledons, whatever food material is stored inside it, and show the further growth. Later on, what would happen? The leaves would develop, and these leaves would now do, in the process, entire cotyledon would be consumed. These leaves would do photosynthesis, and the plant would meet its nutritional requirements by the process of photosynthesis. So this is the entire process of seed germination. Now I have to deal with the term stratification. What is stratification of seed? We were talking about dormancy and quiescence. Quiescence can be broken down as soon as favorable conditions are given. What about if the seeds are dormant? If the seeds are dormant, they have to be stratified, and the process is known as stratification of seeds. Now, what is there in stratification of seeds is that a seed is taken and then it is kept in the moisture, the moist seed. It is subjected to oxygen availability, and then it is subjected to variable range of temperature differences. Alright, variable durations of temperature would be taken into consideration, and either high temperature or low temperature would be given to the seed so that the dormancy is broken. Any of these conditions which are present, that could be overcome by the process of stratification. Alright, so what is stratification? Stratification is basically breaking down of dormancy. The dormant condition would be broken down by subjecting the seed, moist seed, in the availability of oxygen to variable durations of the temperature. Either that could be high or it could be low. So the stratification of seeds would lead to seed germination if the seed is dormant. Otherwise, if the seed is quiescent, in certain conditions, as in the case of your sprout salad that you eat, you just simply put the sprouts inside the water, and within 2 days you get the seeds sprouting. What is that? That is basically seed germination. Those quiescent seeds that were present inside your packet of the sprouts, they get the moisture and water, moisture and oxygen, I'm sorry, they get moisture and oxygen and they germinate. That hilum thing is broken down, radicle comes out, radicle and plumule would come out as soon as they are kept inside the soil, and a new plantlet would be born. Okay, now what are the factors which are responsible for bringing about this seed germination? Now come back to the stratification point of understanding. It said moist seed, it said oxygen, and it said temperature. 3 things I used in making you understand what is stratification of seeds. So these would be the factors which would be affecting the seed germination. First one is water availability. Of course, moisture is very, very important, because imbibition would take place in the presence of a solvent that would bring about the breakdown of the seed coat through which the radicle and plumule would come out. Oxygen availability is necessary because all the physiological processes, that is the breakage of polysaccharides and their breakdown and their metabolic processes that follow henceforth, they are to be conducted in the availability of oxygen. In certain plants, light also plays an important role in germination. That is the red region of light in case of tobacco and few varieties of lettuce that we consume as a salad. In tobacco, that is a fumigatory substance consumed worldwide in the form of cigarettes and other substances. These plants are showing germination in the presence of light. It has been observed. So these 3 factors are important for the germination of a seed, and we overcome the quiescent or dormant stage in the seed and a new plant